this picture shows kind of zoomed out and then zoomed in as well. So remember, DNA molecules are big. And in the DNA molecule, there could be many, many genes. So we're just going to look at one gene. How does one gene get turned into one polypeptide? Well, the DNA has this codon triplet code that we just talked about. So here's how it works. Every 3 nucleotides is like one genetic word. These are called codons. So AAA, that's the first codon. CCG, that's the next codon. GCA, AAA. So altogether, we have 4 codons in this DNA. Now, during transcription, the job is not really for the RNA to understand the DNA or anything like that. There's no real understanding of this code. Basically, the RNA is just it's the same message as the DNA, but it's written in the RNA language. So all of these RNA codons are going to be complementary to the DNA codons. Remember the base pairing rule. A pairs with T, C pairs with G. But with RNA, we don't have thymine, we have uracil instead. So A pairs with U. But if there were any T's in this DNA, they would still pair with A in the RNA. So during transcription, we turn this DNA codon sequence into a complementary RNA codon sequence. UUU is complementary to AAA because of the base pairing rule. GGC is complementary to CCG, and so on. Now, during translation, this RNA message is going to be read with the help of ribosomes and also tRNA molecules. And we're going to use this information to build a string of polypeptides, okay? We have many peptide bonds. So we're building this string of all these different amino acids. Now, how do we know which amino acid to get? Well, the RNA codons actually tell us which amino acid we need to go get. So you can see here how the DNA code is storing information that will eventually tell us exactly which amino acids need to be put together. This is the information, and this is what we're trying to build here, is the actual amino acid sequence. So the genetic code, the DNA, just like we've been saying all year, it's information, and the information is how to build a polypeptide. So we can see here, this information, this triplet code, eventually codes for a string of amino acids. Basically, every single living thing that we know of does this. They use this DNA code to make polypeptides. This is universal in life. And there's different types of codon charts you can use to figure out, well, which amino acid are we going to build? This one's pretty popular, this circle. I kind of prefer a different type of codon chart that looks like a table. I'll show you that one later, but a lot of people like this one. They think this one's a little easier. So either one is fine. But basically, what we do is if we look at the code, so for example, let's look at this code here. UUU, okay, which was the first RNA codon here, UUU. So now we'll go to our codon chart. We'll start here and we'll go to U. Okay, and then the next letter is U, and then the last letter is U. So we know that this codon here, is going to code for an amino acid called phenylalanine. So that means that this little square here is a phenylalanine. Let's do the next codon, GGC, and we'll go to our codon chart. G, G, C, glycine. Okay, so now we know that this little circle is a glycine. So you can read the RNA codons and it tells you exactly which amino acid that we need to look at. CGU, CGU, arginine. So now I know how to read the RNA language. We have phenylalanine, glycine, that one I think was arginine, and then we have another phenylalanine here as well. And we can actually get some practice doing this in the next video here.